Hello friends, welcome to my YouTube channel. Today we are going to discuss about plasma membrane. Plasma membrane is an externalmost boundary of cell. It covers cytoplasm. As you can see, we draw the diagram of bacterial cell, plant cell and animal cell. You can see the outermost covering of plant cell and bacterial cell is cell wall. But there is no process which is covered under the cell wall. But plasma membrane act as a permeability barrier. It transports the material from inside to outside and taken out material from outside to inside. Chemical composition of plasma membrane. We consider RBC's plasma membrane as a model organism, model cell. It is made up of protein, lipids and carbohydrates. We study RBC's plasma membrane because it doesn't contain any cell organelle. The composition of RBC cell. It contains protein which is 50%, lipids which are up to 50% and carbohydrate which are up to 1%. Now we see carbohydrates of plasma membrane. It is present in plasma membrane. It has very low concentration up to 1%. It never present in free form. It is either attached with proteins or lipids. If carbohydrates are attached with proteins, then it is called glycoprotein. And if carbohydrates are attached with lipids, it is called glycolipid. Carbohydrates always present in extrinsic phase. As you can see in diagram, carbohydrates are present in extrinsic phase attached to lipid and carbohydrate, which are glycolipid and glycoprotein. Lipids. Lipids are the esters of fatty acids with polyhydroxy alcohol. Ester bond present between the fatty acid. Polyhydroxy alcohol. It is classified into two types, which is glycerol and sphingosine. Glycerol is 3 carbon atom compound, and sphingosine is 18 carbon compound. As you can see the structure of glycerol, which has 3 carbon atoms, and the sphingosine, which has 18 carbon atoms. Fatty acids. Fatty acids are of two types, saturated fatty acids and unsaturated fatty acids. In saturated fatty acids, carbon-carbon single bond is present, which is trans in nature, means it is in straight line either. But in unsaturated fatty acids, carbon-carbon double bond is present, which is in cis form, means it is in bent shape. Second example is phosphatidylcholine, which has phosphorus attached to the CH2 carbon bond, and there is presence of choline structure. Phosphatidylserine, which has phosphate attached to the carbon third bond and serine structure is attached. Inositol, this is sugar inositol, which is attached to the oxygen and the phosphorus bond, which is attached to carbon third bond. The PE, PC, PS and PI, all phospholipids synthesized in endoplasmic reticulum, smooth endoplasmic reticulum. Two fatty acids obtained through glycerol based phospholipids. Sphingosine based phospholipid, for example sphingomyelin. As you can see the structure of sphingomyelin, the sphingosine plus attached to fatty acid is considered as ceramide. It is synthesized in Golgi. And then we can see the glycolipids. They are ceramide basic structures. One is cerebroside and other is globosides or gangliosides. In cerebrosides, ceramide is attached with simple sugar, whereas in gangliosides, ceramide is attached to the complex sugar oligosaccharide. Thank you.